Hello, my name is Aryan Kurniwan along with Madison Kahn from the University of Windsor, and thank you for virtually attending our presentation titled The Influence of 4-Period Duration on the Preparation and Control of Sequential Aiming Movements. When movements are performed to two targets in a sequence, reaction time is typically longer compared to single target movements. This one target advantage is contingent on advanced knowledge of the number of targets. Likewise, movement time to the first target in a two-target sequence is longer compared to a single target movement. According to the movement integration hypothesis, segments are loaded into a buffer prior to response initiation, which then enables the second segment to be implemented during movement to the first target. The movement constraint hypothesis is based on the assumption that variability in limb trajectories to the first target are constrained in order to meet the accuracy demands at the second target. In studies that have shown the one-target advantage, the 4-period, or time prior to the stimulus, has typically been varied randomly. Although the one-target advantage depends on prior knowledge of the number of targets, there has been no systematic investigation of how the one-target advantage is influenced by 4-period duration. As shown in the current study, this has important theoretical implications. To test if the one-target advantage is influenced by 4-period duration, we performed two experiments. Participants were seated at a table with a touchscreen laptop placed on the tabletop. Targets were presented on the screen and participants performed aiming movements using a handheld stylus. The position of the stylus was tracked using an opto-track system. Participants were required to perform 1 and 2 target aiming movements. At the beginning of each trial, the participants were required to align the tip of the stylus on the start position. Once the stylus was steadily aligned, the targets appeared. Following a variable 4-period of 0, 500, 1000, 1500, or 2000 milliseconds, the stimulus was presented, which consisted of a tone. In the one-target trials, participants were required to lift the stylus from the start position and touch down at the first target. In the two-target trials, participants were required to move to the first target and then continue their movement to the second target. Participants were instructed to move as quickly and accurately as possible. In Experiment 1, the 1 and 2 target test trials were administered in two separate blocks. The 5 4-periods were randomized within a block of trials. In Experiment 2, the number of targets and 4-periods were varied randomly from trial to trial.
Results. Similar to past research in which the number of response segments was known in advance, reaction time was longer in the two compared to single target responses at all 4 periods. While the reaction time difference between 1 and 2 target responses was not influenced by the 4-period duration, the 1 target advantage in movement time did increase in magnitude as the length of the 4-period increased. This finding supports the movement integration hypothesis by showing that sufficient time is required to load segments into a buffer, which then enables the second segment to be implemented during execution of the first segment. The current results are also consistent with the movement constraint hypothesis. Variability at the end of the first movement was significantly less in the 2 compared to 1 target condition. The results of Experiment 2 revealed that reaction time was longer in the 2 compared to 1 target condition only at the longer 4 periods. While the 1 target advantage in movement time did not emerge at short 4 periods, movement times to the first target were longer in the 2 compared to 1 target condition at longer 4 periods. These results add further support to the movement integration hypothesis. There was no difference in variability of the movement endpoints at the first target between the 1 and 2 target conditions. Hence, there was no evidence to support the movement constraint hypothesis when target conditions were randomized. Discussion. Collectively, these results support the movement integration hypothesis by demonstrating that planning and execution of sequential aiming movements is influenced by the interval prior to the stimulus under both blocked and randomized trial sequences. Support for the movement constraint hypothesis was revealed only when the number of targets was the same from trial to trial. It appears that visual regulation plays a dual role in two-target sequential aiming movements. First, along the lines of the movement integration hypothesis, the execution of the first segment is visually monitored in order to optimally time the implementation of the second segment. Second, along the lines of the movement constraint hypothesis, visual regulation serves to adjust limb trajectories to minimize variability at the first target. Reducing variability at the first target not only ensures that accuracy demands at the second target are met, but also enhances the transition between segments.